Hello everyone, welcome to this new series by Drishti IAS, which is related to your Prelims 2026. And in this video, we are covering our 100 most important topics related to your science and tech. So let's start the video. So the first one is your vaccine platforms like your mRNA vaccines, viral vector vaccine, DNA vaccine, and inactivated vaccines. And we want to know about the comparative mechanism about these vaccines. So this is the major topic that we need to know. After that, there is a microbiology of the zoonotic disease. So microbiology means your virus, bacteria, fungus, protozoa, which will be the causative agent of any disease. And also we want to know about this word which is come in front of us after this corona. This is your zoonotic disease, very talkable topic. And inside of it, we want to know about the fundamental differences, virus, bacteria and fungi. And we also want to know about the Nipah, Zika and Ebola, which is very talkable topic. After that, there is an antimicrobial resistant. This is a big problem. WHO already say that this can be happen with us, and if it can be regularly going on, this can be new pandemic in the world. So antimicrobial resistant. And we want to know about the mechanism for the resistant, superbug, which is the name of microbes which can develop your resistant, and also what is the global health framework for the protection of this problem. So this is the thing that we are going to talk about in this video. So let's start. So the first thing, mRNA vaccination. So actually before the corona, we have a basic method like we have inactivated vaccine method, toxoid based vaccine method, or maybe we can say live attenuated vaccine based methods. But now there is a new concept can be come in front of us. This is called as mRNA. And you might be know about this M word. This is related to the messenger RNA. It means we want to put a RNA inside our body in the form of vaccine. And this RNA is basically a type of message which can be given to our body for our immune system, that you can read this RNA structure, and this can be related with any pathogen which can be come out in future into your body, and you have to prepare your antibody against of it. So this kind of the medicine can be ready for mRNA vaccination. So what is the mechanism? So this vaccine contained the messenger RNA, a small part of your mRNA, which including viral antigen, usually spike protein, like your coronavirus have the spike protein. So this is basically a small part of RNA which is comes from any viral antigen, or maybe we can say any pathogen. So you can basically put it inside our body. After that, mRNA enter host cytoplasm. So it can be entered into the cytoplasm, ribosome translate it into the antigen. So when a virus can capture our body cell, the whole mechanism of protein synthesis is captured by virus. And due to that, there is a spike protein formation can be done by ribosome. And this activity against the body function, and then our immune system become active against it. So example, there is a no entry of nucleus because we don't use the DNA or maybe whole cell. There is only mRNA, which is only the part, and this is degraded naturally, because when your body make the immune system against antibody for it, then these antibody kill this mRNA which you put by the vaccine. Like America made the vaccines related to mRNA based platform, the Pfizer one and Moderna, both are related to your corona, because this technology is come in front of us after the corona. So American, USA based Pfizer and Moderna is based on mRNA vaccination, very high attacking power, and we can say very high preservative power against the coronavirus. What are the advantage? It can be rapid design and scalability. We can produce in very high in manner. There is no risk of infection because very small part of RNA is used, and there is a high immune response is there. So your immune system become very active for it. And what are the limitation behind it? So they require the ultra cold storage. This is the big problem. If I tell you about the storage problem, so minus 60 degrees Celsius to minus 90 degrees Celsius, this kind of the minimum temperature is required for storage of this vaccine. So before the build-up of this vaccine, we need to build the storage capacity. And with India, we have the capacity to develop mRNA-based vaccine, but this is the problem for storage. And when this kind of the storage can be happen, so this can be stored in one place, but the transportation of this vaccine can have a big problem. So mRNA is thermally unstable, because if you can lose this temperature, or maybe you can say they can get the high temperature heat, that can easily destroyed. So this vaccine need to be preserved in very low temperature. Cryopreservation basically needed. Clear? Next one, viral vector vaccine, and the name is very simple, viral vector. It means you are using virus as a vector for vaccination. So what is the mechanism? We use a harmless virus, means we can minimize the infective percentage of a virus and the infective ratio is become degraded. Then you can use it by engineered carry antigen gene. So vector infect the host cell, because if you can enter the live virus, so live virus can be entered and it can attack on a cell, host cell. This cell make antigen gene expression, and by triggering this point, we have to active our T lymphocyte cell. It means we put a live virus into our body, and this live virus can attack a cell, capture it, making the bad protein, or maybe we can say useful protein for itself against the body function, and then this process is under T lymphocyte coverage, so our immune system become very active for it. Type, they are non-replicating vector, most common, because we put the whole virus into it, and replicating vector, which is rare, means less virus can be replicated by directly, and they are basically infected by the cell, and lots of the cell can be infected by again and again. Examples, the Oxford, AstraZeneca, Johnson & Johnson and Sputnik V, they all are using live attenuated, which means weak virus, and our Indian vaccines is also made by this part, but there is only one difference. AstraZeneca, Johnson & Johnson, Sputnik V, they are using live virus, weak virus, but in India, Covaxin and Covishield is made by dead coronavirus, inactivated virus basically. This is the only difference. So mRNA vaccination for corona, that is made by USA, Moderna and Pfizer. Sputnik, Johnson & Johnson, AstraZeneca, Oxford, they are making live attenuated virus based vaccine. When we talk about the advantage, this can be stable between 2 to 8 degrees Celsius. So low temperature is needed, but normal one. A strong cellular immunity, because your immune system can fight against a live virus. There is a limitation. So pre-existing immunity against vector may reduce the efficiency. Maybe your body can make a different type of antibodies, and that antibodies attack on this virus. So again your system cannot be activated in that manner. So maybe less immunity can be happen. So partial disease in life that can be destroy the function of this vaccine. This is the problem with it. And rare adverse events are there. So this is a good vaccine. When we talk about DNA vaccine, so we use the direct DNA with it. Name is simple. So you can use the plasmid DNA. You can know that plasmid DNA, which is known as your extra nuclear DNA. So basically you are using the extra nuclear DNA. DNA enters into the nucleus. This is the major point, and transcribed into mRNA and antigen. Because if nuclear DNA is changed, this one form the new mRNA, and new mRNA produce the new type of antigen protein, which is basically act against the body. It require delivery system like electroporation, or maybe we can say which is a needle free injectors. So we cannot use the needle for it, but we have to use the delivery system differently. There is example Zycovir-D, and this is the new type of vaccination which can be basically given by nasal based. So you can just sprinkle it into the nose and you can inhale it by your respiration system. This can be under the trial, available but under the trial. Lots of the issues is related to it. and the issues is related to it is your delivery challenge, how you can provide it. So advantage, highly stable because no storage is required, easily storage and transport is there, and no live pathogen. So we cannot use the mRNA, we cannot use the virus, that's why it is the most safe method. We talk about the limitation, lower immunity, so it needed the booster. You know the booster dose? When we use vaccine for limited time period, and again you need one more vaccine for limited time period, as you done for your Covaxin and Covishield. So with the time period you need the booster dose. This is the problem with it. And also delivery challenge. This is the best problem related to it. And that's why I can say that delivery challenge is the big thing. That's why we have to be under trial. It is developed, but delivery challenges creating the problems. Now, the next one, this is our Indian made inactivated killed vaccine. It means inside of this vaccine, we use the dead coronavirus. So dead coronavirus is used. So use chemically or heated inactivated whole virus, inactivated dead virus. It cannot replicate because dead virus retain antigen structure, just a structure that we provide the body to read. So primarily introduced antibody and mediated immunity. So they can produce the mediated immunity. Your Covaxin, Syncovac is there. Covishield is also related with it. So what is the advantage? Well established technology. Already we are using it. Safe immunoresponse is there. Easy storage. No need to be develop a different kind of infrastructure for the storage. And weaker T lymphocyte response, because why weaker? Because your virus is already dead, so our immune system cannot take it seriously. So they active, they killed it, they remove it, but not too much because the cell is already dead. So T lymphocyte cannot have a function to fight, so they are having a weaker T cell response. It required the adjuvant and boosters. So with the time period you also do some booster dose with it also. That's why you need the booster for your Covaxin and Covishield. If I am talking about the fundamental difference of virus, bacteria and fungi, three major things, because we already done with the virus, because virus is related to your vaccine, and now three different type of pathogen. First, virus. It is a acellular, not true living organism. You know that virus is considered as a connecting link between living and non-living. So genetic material can be DNA and RNA, and this will be the more important point. We can simply say virus is the only structure in this living world which can have both DNA and RNA as a genetic material. But both DNA and RNA cannot be present together. They are always present separately. So a virus can have DNA or maybe have the RNA. But RNA as a genetic material can be only present in virus. Rest whole living world have the genetic material DNA. Simple as that. So when we talk about it, it is obligate, intracellular, parasite, replicate only inside the host cell, because they are doing the living activity inside of the living cell. There is no ribosome, no protein formation, no metabolism. There is no their own activity because it cannot have any kind of the cell organelle. And there is no effect of antibiotics, because antibiotic had the word biotic, means life. So antibiotic is the medicine which is against the life, and this one don't have the life. It is a abiotic structure, and that's why we can say antibiotic medicine cannot have any effect over the virus. Influenza virus, HIV virus, Ebola virus, these are the examples. Now we talk about the most common bacteria. So bacteria is the example of prokaryotic cell, undeveloped, unicellular. Genetic material is DNA only. As I tell you, RNA as a genetic material can act only in virus, never be with the other one. All have DNA. So next one, have cell wall, peptidoglycan, and we also called it muramic acid. What we called it? Muramic acid. So we also called it muramic acid. There is a presence of ribosome. That's why bacteria can make protein. It can live independently, and it is also affected by antibiotic, because biotic means life and this one have the life. So against the life antibiotic, like tuberculosis for TB, this is Mycobacterium tuberi, tuberculosis disease, cholera, Vibrio cholerae is there. When we talk about fungi, so fungi have only one major point, that is the outer layer made by chitin, which is a polysaccharide. It is a eukaryotic organism, but the cell wall is made by chitin, like unicellular yeast and multicellular molds, which can be present with our food infection. They can reproduce by spores, means small cell-like structures, and antifungal drugs are used for the treatment of fungi, like Candida aspergillus. So this is the fundamental difference. Now, very interesting topic is known as your zoonotic disease. Very interesting topic, zoonotic disease. Actually, this word is come in front of us after corona. So zoonotic disease means those category of disease which can be transmit to human body by any kind of the animal. Means our natural system, our habitat can have different type of domestic animal, or maybe we can say other animal. But we are affected by any kind of the disease, and that can be transmitted by any animal. Like very simple example, malaria is transmitted by mosquito Anopheles. This is also zoonotic disease. And when we talk about the plague, this is also infected by rats, which is present in home. Rabies, cat, monkey, dogs have this kind of the virus. So when a infective disease can be present in human body, but the infection vector is always a animal. So animal to human body transition of disease can be noted under zoonotic disease. And this will give you a new type of health structure mechanism, like if you want to protect a human, you have to protect our animal also. Because if a virus can attack on animal relationship with human, then they can be transferred to the human body. So you have to protect our animal. And why only animal? There is some plants also, because plants are also present near to us and they can also use as a vector for transferring any kind of the disease. So there is a whole concept can be come out after this zoonotic disease concept. If you want to protect human health, you have to protect the environmental health, means plant health, animal health, and after they both get healthy, the human also get healthy. So this is the zoonotic concept. So basically the definition, which is the disease transmitted from animal to human, like any kind of the event can be there. No, we can say only one event, but actually any kind of the impact. Animal to human transfer of the disease. And if we talk about this one, the first one example is your Nipah virus, very talkable. So basics is there, and this one is a virus family. So para myxoviridie. So para myxoviridie is the family of it. Natural reservoir, fruit bats. So fruit bat is the natural reservoir which can have this virus. So this is the animal. And when we talk about transmission, so it can be transmitted from bat to human, and then again it can be transferred from human to human. So bat to human, first time contaminated fruits, if you can eat any kind of the contaminated fruit, so bat to human, and then again it can be transferred from human to human by the body fluids like blood, saliva, tears, sweat, urine, body fluids. Key features, there is a severe encephalitis. Encephalitis means brain swelling is there. Very high fatality rate, it means it can be increased very rapidly, so the people can get infection very high in manner. And recurrent outbreaks in Kerala. So Kerala is the one of the place which can have highest amount of infection of Nipah virus. Vaccines, presently there is no licensed vaccine, so experiments are going on. When we talk about the next one, Zika virus, this is belongs to the family of, this is again virus, Flaviviridae family is there. Vectors, Aedes mosquito, which is present near to us, domestic mosquito, and this can be transmitted by mosquito bite. So again zoonotic disease. Sexual transmission is there, this one is the most important point. Like if someone asks you that Zika virus can be infected by sexual transmission or not, yes, and UPSC already asked this question. So Zika virus also have a sexual transmission, and this can be transferred from mother to fetus. This can be very, very highlighted thing. This can be transferred from mother to fetus. During the pregnancy, it can also transfer. So what are the key features? Mild fever is there, so the people cannot be get serious for it easily. It is caused by microcephaly in newborns, means they are creating the problem of development of baby. Also linked with Guillain-Barré syndrome. This is again a big problem that is comes after the corona. So Guillain-Barré syndrome is like that when our nervous system can face some problem. Like you know that our nervous system have one part which is known as your PNS, means your peripheral nervous system, which is related to your nerves. And nerves are basically those structure which is responsible for carrying and transferring the signal from brain and body. So this kind of the Guillain-Barré syndrome, your brain is active, they can receive the signal, they can have the capacity to send the signal, but the signals cannot be properly initiated in the body because the nerves which is present in overall the body, basically PNS, peripheral nervous system, have to be impacted, and this one cannot be active for properly receiving and giving the signal. So the some people can face many kind of the body paralysis thing, but that can be limited for the time period, and this kind of the symptom can be present for 3 months, 6 months or 1 year, or maybe for the 2 year, and after that our immune system get recovered easily and we can get again it. But if between that time period you can get the infection of again any kind of the disease, this one creating the permanent paralysis of the body. So we can say no approved vaccine is there. So there is no approved vaccine right now. This is the big problem. And already we know that making a vaccine for virus, this is again a very tough task. Clear? Next one is your Ebola virus. So the family is again same, Flaviridae. Reservoir is your fruit bat again, fruit bat, named after Ebola river in the Africa. So this is the another point. Transmission by contact with infected animal, though this one again transfer from human to human. So infected animal transfer it to human first, and this human act as a vector and basically send the disease to the other one, like blood and body fluid. There is a hemorrhagic fever, which means high intensity of fever is there. Hemorrhagic, very high intensity, very high mortality. So this can be increase their effect very rapidly, and also it can harm other person very fastly. So cause multiple organ failure. This is the problem related to it. Multiple organ failure. This is the problem. Vaccine is approved and used during the outbreaks. So vaccine is approved for your Ebola virus. Now, there is a problem which is known as AMR, because there is a two kind of the words come in mind for the treatment. The first for virus, that is your vaccine. We know the vaccine type, and there is a second thing, antibiotic medicine. So antibiotic medicine is used for microbial treatment, means for bacteria disease, for protozoa disease, for any kind of the fungal disease. But the problem is this, कि in India and in world, but in India, lots of the person can use antibiotic medicine very easily. They cannot go to the doctor, they cannot get the prescription, and they can simply get the antibiotic medicine from any medical store and they can easily consume it. And with this consumption, there is a rapid use of antibiotic can become in mind. And when you can use antibiotic medicine for multiple time, so your body and antibiotic infection can be there. So your body have high amount of antibiotic medicine, and now this bacteria, fungus or protozoa can develop a fight against this medicine. So they can develop the resistant, and due to that unusual use of antibiotic medicine can produce the resistant in microbes. So basically now you can get a disease by any microbe and you can use the antibiotic, but that microbe don't have any effect of this antibiotic medicine. So there is a big problem for the treatment. So this is creating a huge difference in medical industry. So when we talk about AMR, so antimicrobial resistant, when a microorganism like bacteria, virus, fungi and protozoa evolved the ability to survive against the drug which is designed to kill them. So they have development of this kind of the activity. It leads to treatment failure, longer illness, higher mortality and economic burden. When we talk about the mechanism, so there is a first thing, misuse and overdose of antibiotic medicine in the human, self-medication without any prescription, over-the-counter availability, incomplete treatment course, like doctor can say use this medicine for 5 days and you get the positive result after 3 days and you can just leave it for 2 days. This kind of the incomplete treatment, use of antibiotic for viral infection, because you already know that virus cannot have any effect for antibiotic medicine, but the people can use it like your cold and flu. Excessive use in poultry and livestock. So antibiotic medicines are also used to the growth of promoter. So they can basically used in plantation, increase the size of vegetable, freshment there, and this can be consumed by us and we get the antibiotic medicine. So poor infection prevention and control, like overcrowded hospitals. So the doctors want to be go the empty bed process, or maybe we can say the doctor wants to empty the bed very early so they can give you the antibiotic medicine. You get a better feeling easily in very short time period and you just go to the home. So doctors can also use it badly. So this is overcrowded hospital can make this process very rapidly, or maybe we can say this is again the reason for utilization of antibiotic medicine. And also there is a inadequate sanitization, or maybe we can say hygiene. So when you cannot use the hygiene method, you can easily get the infection from bacteria or anything other. So you have to use multiple medicine in short time period. There is an environmental contamination, like pharmaceutical waste in the water body, and you can use this water body. Antibiotic residues in the soil, untreated hospital and animal waste. Untreated hospital and animal waste. So this is basically again present with our food system, water system. So we again consume the high amount of antibiotic medicine. There is a weak regulation and enforcement. Easy access to the antibiotic without any doctor prescription, you can easily get it from medical store. Poor monitoring for the prescriptions, no one can use this kind of the thing. And substandard of drugs. There is a globalization and travel. This is again a big problem, big concern. Like international travel spread the resistant strain quickly, because if you are getting the infection, you can move somewhere other countries and you can spread the disease easily from human to human. There is a medical tourism contribute to the global spread, because medical tourism is like that you can get the medical services in other country by this, and by this an infected person can be transferred to the other place. This can also be spreading method. There is low awareness, or we can say poor health literacy, because the people don't want to know about it, that when antibiotic can be used or when cannot be used. Already government can use one rule that is called red data line, and basically they can mention a red line in antibiotic medicine, that they can request you that before purchasing any kind of the antibiotic medicine, you can see that red line, and if it is mentioned in that part, you cannot purchase it without any doctor's prescription. But the people cannot be have this kind of the knowledge and literacy. Next part, there is a slow development of new antibiotic medicine. So we want to develop new one, but this is having a very slow process. Next one, what is the meaning of superbug? So actually AMR, antibiotic microbial resistant. This one develops such a microbe which cannot be impacted by antibiotic medicine. So actually we named it superbug, which cannot be killed by antibiotic medicine that we have right now. So the pathogens resistant to multiple or all available antimicrobial medicines. Important examples are there. MRSA, XDR-TB, this is one of the most important example, and also there is a NDM1, you might be know about it, New Delhi Metallobeta. New Delhi Metallobeta. So this is a bacteria which is present in water supply of the Delhi, and this can create the digestive problem, and when you consume the medicine this cannot be defected because they have the resistant against it. So lots of the examples are there. Extra drug resistant TB, MRSA is there, CRE is there. So WHO classifies them as a critical and high medium priority pathogen because they are such a problem which can creating a big issues in the world. Next one, what is the global structure? So WHO, World Health Organization, AMR recognized as a global public health emergency. It leads the coordination and surveillance. Global antimicrobial resistant surveillance system is there, which is known as your GLASS, and this organization track the AMR trained across the countries. India is a participating member of this GLASS, means you have to track the AMR records. There is a alliance between the multiple organizations, WHO, FAO, WOH, UNEP, which promote the One Health approach. And as I tell you, if you want to be healthy for the human being, we all want to be healthy for animal and health method for environment also. And this is known as One Health system. We talk about it more. So One Health approach. So there is an integration of human health. There is an integration of human health, animal health, environmental, global One Health framework. So we want to maintain a health framework which is available for human, animal and plantation means environment. So this is the One Health approach. Leading institutions, which institutions are doing this? WHO, World Health Organization, Food and Agriculture Organization, World Organization for Animal Health and United Nations Environment Program. So environment is there, health is there, agriculture is there, and WHO for human health. So jointly promote the One Health governance, surveillance and the capacity build-up. This is known as your One Health approach. India want to start it from 2030. Like we have our national health policy right now, and this health policy is started in 2017, and that will be the time period for ending 2025. But due to corona time period, we have to be extend this process, and now currently we have the old one, but after 2030 we start our new program for health framework. This is known as your One Health program. So after 2030 we have to plan to start this. There is a national One Health mission started in 2022. It is launched by the government of India. Integrated disease for surveillance, zoonotic disease preparedness and AMR contaminant. So these are the topics that we are covering under the National One Health Mission. This is the preparation for starting of One Health system in 2022. There is an Indian Council of Medical Research, ICMR, which coordinate for human health surveillance, and also AMR research and pathogen tracking is there. Indian Council of Agriculture Research, ICAR, it monitor the animal health. It monitor the animal health and veterinary drug use. Means animal used medicine concerns are there. So ICAR basically looking for the veterinary drug use, because excessive use in animal that can also leads the AMR condition. Next one, One Health Support Unit that is present in Pune, National Hub for the cross sectorial coordination. Why the One Health is important? This one is a important part for your prelims. Why it is important? So 75% of the emerging infections disease are zoonotic. 75% disease can be come from animal. There is a deforestation, climate change, increase the human animal contact, because if the forest can be less, the animal can be more easily connected with the human, because they also live with us in near about the habitat, and this is creating a big problem. AMR is spread through the human, animal and the environment. That's we include all of them in One Health. There is a pandemics like Ebola, Nipah, COVID-19, and they all are come from animal, means they all are zoonotic disease. That's why One Health approach is more important for us. Next one. So now this topic is over and I will provide you some questions for this. So this is your PYQ. So let's talk about that. First you read the question, then we go for the answer of it. So the first question related to it, which of the following are the reason for the occurrence of multi-drug resistant in microbial pathogen in India? So what is the reason? What is the reason behind it? Why the AMR can be happen? Genetic predisposition of some people, taking incorrect dose of antibiotic to cure the disease, using antibiotic in livestock farming, and multiple chronic disease in some people. And already we know about it कि yes, the disease can be multiple time can be happen with the people, so the people can easily use the antibiotic medicine many time. Use of antibiotic medicine for the farming, this is again the problem. Taking incorrect dose, this is again the problem, but there is no genetic relation. Antibiotic medicine does not have any genetic relation. Clear? So the question, second, third and fourth statements are correct. Second, third and fourth, your correct answer is D. Your correct answer is D. Next one. With reference to the recent development regarding recombinant vector vaccine. Recombinant vector vaccine, so basically it is rDNA vaccine. Recombinant DNA vaccine which can be made by genetic engineering. So which statement is correct? So genetic engineering is applied in the development of these vaccine. Yes, you know that. Genetic engineering is applied for the use of recombinant DNA formation. So the first statement is correct. Bacteria and virus are used as the vector. Yes, they are the vector. We know that vaccination formation can be done by bacteria and virus. So the second statement is also correct. So C option is correct. Next is your again PYQ. Consider the following statement. In tropical region, Zika virus disease is transmitted by the same mosquito that transmit by dengue. Yes, we know that dengue is transmitted by Aedes mosquito. Aedes mosquito, and the same is used for Zika virus. Sexual transmission of Zika virus disease is possible. Yes, it is possible. As I tell you, UPSC already asked this question. Clear? So both statement is the correct one. C option is the correct one. So this is your PYQ. This is again a PYQ. In the context of vaccines manufactured to prevent the COVID-19 pandemic, consider the following statement. The Serum Institute of India produced the COVID-19 vaccine named Covishield using mRNA platform. So you all guys know that Moderna and Pfizer is the only vaccination for COVID made on the behalf of mRNA. Our Indian have inactivated virus for it. So this one is a wrong statement. No mRNA is used. Sputnik V vaccine is manufactured using the vector-based platform. Yes, you know that there is a live attenuated virus is used. We know it. So this is again the correct statement. So first is wrong, second is correct. Covaxin is an inactivated pathogen based vaccine. Yes, this is the correct statement. So second and three are the correct. B option. Clear? Very easy one. After reading this topic, you are capable to know about the answers. Now, this is your practice question and I have 1, 2, 3 questions for your practice. So 3 practice questions. So there is a 3 practice questions for you and you have to read this topic again, watch the video again and again, clear your concept, and then you have to read this question and give your answer after this video in the comment box. So thank you for watching.
For more informative content, like, share and subscribe, and do not forget to press the bell icon to get the notifications.